everyone, and welcome to the Real Science Exchange, the pubcast where leading scientists and industry professionals meet over a few drinks to discuss the latest ideas and trends in animal nutrition. Hi, I'm Scott Sorrell, one of your hosts here tonight at the Real Science Exchange. Tonight, we're continuing the new mini-series of podcasts recorded at the 2022 Cornell Nutrition Conference called New Revelations in Transition Cow Nutrition. I'd first like to welcome, though, my co-host, Dr. Clay Zimmerman. Clay, thank you for joining me here once again. And as always, what's in your glass tonight?
Tonight, being the world-class athlete that I am, I'm drinking my favorite sports drink this evening.
Oh, very nice. Well, good. Well, tonight I'm having something called, so I wanted to have something that I thought Jose would drink, but I don't know what he drinks. So he's from Brazil, so I'm having a caipirinha. And that's it's made with kind of a Brazilian rum and a little bit like a mojito or kind of like a margarita, but these are very tasty. I kind of fell in love with them when I was in Brazil not that long ago. So cheers, Clay.
Cheers.
Yeah.
Tonight's pubcast stories are brought to you by ReaShure Precision Release Choline. ReaShure is the most researched encapsulated choline on the market today, consistently delivering results to your transition cows of higher peak milk, reduced metabolic disorders, and even in utero benefits to her calf, leading to growth and health improvements. Visit Balchem.com to learn more.
So as we begin tonight's discussion, we'd like to note that this is the second in the series of 4 podcasts taken from the mini symposia Balchem sponsored at the 2022 Cornell Nutrition Conference. If you'd like to see the full presentations and slide decks, go to balchem.com/real science and search for the 2022 Cornell Mini Symposia. This podcast features Dr. Jose Santos from the University of Florida. Clay, would you like to give us a quick intro to what our audience is going to see when they look at the presentation?
Yeah, basically, Jose was making a case for choline being a required nutrient in for all dairy cows. So he spent a little bit of time, just giving a little history of choline in diets and so forth, and did a nice job walking through the history of choline in dairy cows, which really goes back to the 1980s, and ended with some very recent research that has been done in his lab to really make a great case for choline being a required nutrient for all dairy cows.
Excellent. I look forward to the presentation, and I think you'll enjoy it as well.
I hope that I will present to you and make the case that choline fits those criteria, and it is a required nutrient for dairy cows. So during this New Revelations on Transition Cow Nutrition Symposium, I'm going to spend the next 40 minutes discussing with you some aspects of choline nutrition for dairy cows. So in my presentation, I'll give you a very brief history on choline, the role of choline on intermediary metabolism, then some new data on the effects of choline in reducing fatty liver in dairy cows. I'll spend a little bit of time talking about how choline and what are the impacts of choline supplementation during transition and subsequent productive performance and health. And then I hope to make the case that every cow really, or most cows, respond to choline. And the example I'm going to use is body condition score in response to choline. And then I'll finish up with some potential mechanisms. And I hope by presenting this data, I'll make a strong case that choline is a required nutrient for ruminants as it is for other species. So choline was discovered, the first studies in the 1700s by chemists and pharmacists, but it wasn't until about 40 years ago that work in England by Derek Lindsay here, who showed that most phospholipids in ruminants are synthesized de novo. And the reason for that are synthesized de novo from choline that's being synthesized primarily through transmetallation reactions. Okay, And the reason for that is because most dietary choline that's present either in feeds or if we were to supplement as salts are degraded by the gastrointestinal microbes. And this was then clearly demonstrated by Dr. Richardman at the University of Maryland, who confirmed those findings and showed that more than 90% of the choline in feeds do not show up past the rumen. So they don't show up in the small intestine. Choline or those ingredients containing choline don't deliver choline for absorption in the small intestine. So this required the development of rumen protected products. And upon the development of those products, then numerous universities in the US and in Europe conducted experiments primarily with transition cows to evaluate the impacts of supplemental choline as rumen protected choline on performance and health of dairy cows. Now, choline, I think it is a required nutrient because it's critical for synthesis of phosphatidylcholine as well as sphingolipids, which are needed for integrity of cells, either cell membranes in every cell or the integrity of neural tissue. But choline also plays a role in this one-carbon metabolism because choline can be oxidized to betaine, and betaine can donate methyl groups in transmethylation reactions that affect gene transcription. Choline is also a substrate to be acetylate to acetylcholine that is part of neurotransmission. It's important for lipid transport and absorption. So in multiple species, the recommended daily allowances have been established such that for humans, such as Dr. Zimmerman here, the amounts of choline that are suggested to be consumed daily vary according to physiological state and sex, but anywhere between 400 and 550 milligrams per day. In the case of laying hens, depending upon the intake, the amount can be approximately 0.6 grams per day. Young chicks, it's less, 100 micrograms per day, or 100 milligrams per day, I'm sorry. Lactating sows, depending upon the intake, somewhere between 4 and 6 grams per day. But then when we get to the adult dairy cow, there is this question mark. We just have not established the recommended daily amounts to be fed. And I think part of the reason is because the microbes destroy choline. So we have to feed as a rumen protected product, which creates some uncertainty on the exact amount that's delivered for absorption. So that's one aspect. And the other one is that we probably don't have a lot of dose response experiments demonstrating the optimum amount of choline to be fed to dairy cows. But because we don't know the exact amount, it doesn't mean that it's not a required nutrient for cows. Okay. So obviously choline has major roles, but I'll show you some of the biochemistry of the major metabolic pathways involving choline metabolism. So choline, when absorbed from the gastrointestinal tract, can be acetylate in nerve cells into acetylcholine that's important for nerve transmission between neurons or in the neuromuscular junction. Choline can be oxidized to this betaine, which is a 3-methylglycine product, which can donate methyl groups to homocysteine to regenerate methionine. And in this process, there is an interplay with vitamins of the B complex, such as folic acid. And methionine can be a precursor for synthesis of S-adenosyl methionine, which is the universal methyl donor. So during this pathway of resynthesis of methionine and transmethylation reaction, we can have S-adenosyl methionine that can donate methyl groups, and this can be the synthesis can be enhanced by providing choline to be oxidized to betaine. But choline can also be phosphorylated into phosphocholine, and phosphocholine can now combine with CTP to form CDP choline, and CDP choline is substrate to be phosphorylated to phosphatidylcholine, which is the predominant phospholipid in cell membranes. This pathway of de novo synthesis of phospholipid takes place in almost every cell in the body. But there is a second de novo a second pathway for de novo synthesis of phosphatidylcholine through transmethylation of phosphatidylethylamine, or the PMT pathway, in which methyl groups are donated by S-adenosylmethionine to CDP ethanolamine to synthesize phosphatidylcholine. So we can synthesize phosphatidylcholine through donation of methyl groups from S-adenosylmethionine, which can be, the abundance can be enhanced by oxidation of choline into betaine, or we can synthesize phosphatidylcholine through this Kennedy pathway, this de novo synthesis of phosphatidylcholine through phosphorylation of absorbed choline in gastrointestinal tract. So there's two major pathways for synthesis of phospholipids in cells. Now, when cows undergo transition, we know very well that as cows approach calving, there is an increase in nutrient needs because of the growth of the fetus, concurrent with synthesis of colostrum and uptake of nutrients by the mammary gland. At the same time, unfortunately, particularly when cows are in the last week of gestation, dry matter intake decrease in most cows. And combined with the hormonal change that take place in that period, there is a upregulation in lipolytic signals to accommodate the needs of the mammary gland as cows begin lactation. And this results in extensive lipomobilization. Okay, adipose tissue, subcutaneous or abdominal adipose tissue, is mobilized to supply nutrients to the mammary gland, particularly long-chain fatty acids for synthesis of milk fat in colostrum and synthesis of milk fat in milk. But those fatty acids can be taken up by other tissues, including the liver. But if in excess, the liver will find other pathways to dispose of the excess of fatty acids, primarily through ketogenesis, which can export ketone bodies now as an energy source to peripheral tissues, including the mammary gland, for the novo synthesis of short or medium chain fatty acids. But when excess of fatty acids are taken up by the liver, they can accumulate as fat droplets, as droplets of triacylglycerol, which are quite dynamic, quote-unquote, organelles within the liver, not just a storage site. One of the impacts of accumulation of fat in the liver is that it can induce inflammatory response in the hepatocytes. Okay? So eventually this cow might develop some degree of hepatic lipidosis. And unfortunately, it takes time for cows to dispose of those fatty acids. Okay? Because ruminants evolve without the same ability to export triacylglycerols as very low density lipoproteins as other species. But this hepatic triacylglycerol can leave the liver as very low density lipoproteins, which will deliver fatty acids to peripheral cells, including the mammary gland for milk fat synthesis. But when in excess, that can damage the hepatocytes, can induce a pro-inflammatory response. And in vitro studies have shown very clearly that excess triacylglycerol in the hepatocyte can suppress ureagenesis and gluconeogenesis, important functions for the ruminant liver. So this can affect potentially the health of the cow and influence animal performance. So remember that one of the pathways for synthesis of phosphatidylcholine is through transmethylation reactions, okay, through that PMT pathway. And a potential donor of methyl groups is methionine through the action of S-adenosylmethionine. Unfortunately, when cows undergo the transition period, methionine is limited because now there is increased uptake by the mammary gland for synthesis of milk proteins, including synthesis of immunoglobulins that are transferred into colostrum. So this probably limits the supply of methyl donors for synthesis of phosphatidylcholine. So I believe that there is probably an increased need of choline in this period, not only to provide more methyl donors by being oxidized to betaine, but also to provide choline to be phosphorylated for synthesis of phosphocholine and eventually synthesis of phosphatidylcholine. That would be critical at this point. So when animals have deficiency of choline, or when they have extensive lipomobilization, they can develop fatty liver, as in this example here shown in this picture. And obviously, fatty liver is a common phenomenon in dairy cows. Approximately 40% to 50% of the cows in literature have shown at least a 5% concentration of triacylglycerol in the hepatic tissue in the first 2 weeks of lactation. So fatty liver is very prevalent as far as we can tell, and we believe that this has implication to subsequent production and health. For the sake of time, I'm going to show you data only on incidence of disease. But recently we asked the question, what is the association between the degree of triacylglycerol infiltration in the hepatic tissue in the first week postpartum with production, health, and survival? And here is the health data of those cows. And what we found was a linear association between hepatic triacylglycerol and incidence of disease, either clinical or subclinical disease. So we had about 330 cows in which we collect liver tissue on day 8 postpartum, and we evaluate incidence of disease for the first 3 months of lactation. And what we found was that incidence of metritis increased as the hepatic triacylglycerol increased from 2.5% to 7.5%. So if we look at morbidity, we saw a tendency for increase in morbidity in those cows. We saw a tendency for increase in the risk of those cows leaving the herd by 300 days in milk. So increase from 8.9 to 13.7%. And cows that had increased hepatic triacylglycerol concentration in the liver were associated with an increased risk of hyperketonemia and hypocalcemia. And this is subclinical hypocalcemia, and this is hyperketonemia in the first 2 weeks of lactation, So cows that have increased hepatic liptosis are at greater risk of developing disease, which compromise survival, and that has economic implications to the producer. Now, why do we focus on this transition period? Because this is the time when the concentrations of phosphatidylcholine in plasma are the smallest in the lactation. So we can see here that when cows approach calving and during the first 2 weeks of lactation, the concentration of phosphatidylcholines in plasma are reduced in lactating dairy cows. This is why we think this is the optimum time to provide choline to cows to improve synthesis of phosphatidylcholine at this critical time in lactation. Now, we think that choline, by being a building block for phosphatidylcholine, and phosphatidylcholine by being a building block for synthesis of cell membranes, this becomes critical to the ability of tissues to export triacylglycerol. So choline has these lipotropic effects. So here are 2 experiments. The first one from University of Wisconsin, in which cows were subject to a fatty liver induction protocol. So they were limit fed to consume only 30% of their net energy for lactation needs to develop fatty liver. And half of those cows were assigned to a treatment that received rumen-protected choline. Half of the cows remained as untreated controls. And what the authors showed was that in every cow there was an increase in hepatic triacylglycerol content, but this increase was reduced in cows that received rumen-protected choline. So we can see this marked reduction in hepatic triacylglycerol when cows, pregnant prepartum cows, received choline and were subject to feed restriction. Marcus Zenobi here at the University of Florida addressed the same question, but then he asked, what is the response in terms of hepatic triacylglycerol when we increase the dose of choline ion fed as rumen-protected choline? And we can clearly see that when cows were induced to develop fatty liver, the hepatic triacylglycerol increased, but there was a linear decrease in the hepatic concentration of triacylglycerol as the intake of choline ion increased up to 25.8 grams per day in a rumen-protected form. So again, 2 experiments showing that choline has lipotropic effects in dairy cows. So in Marcus Zenobi's work, we collect liver tissue, and then we decide to explore are genes involved in the effects of choline in the hepatic tissue altered by supplying choline to those cows. So let's just look at this heat map here. So if you look at the bottom of the heat map, we see the treatments, which are the doses of choline ion from 0 up to 25.8. Each row here represents a gene that was differentially regulated by the level of choline fed. And as we go from the red to the green, we see an increase in expression of that gene. In other words, an increase in the message to synthesize a particular protein. As we go from green to red, we see a decrease in the expression of that gene. And we can clearly see that genes involved in antioxidant defense of the cell, genes involved in synthesis of phospholipids such as polypoprotein B100, choline kinase, microsomal transfer triglyceride protein, they were all upregulated. They go from red to green as we move from left to right, in other words, as we increase choline ion intake. Whereas genes involved in synthesis of triacylglycerol or reesterification of triacylglycerol, genes involved in acute phase protein synthesis or in pro-inflammatory response, they were down-regulated as cows consume more choline ion. So this tells me that by supplementing choline ion, we reduce hepatic triacylglycerol, and either the increased intake of choline or the increased synthesis of phosphatidylcholine or the combined effect with the reduced triacylglycerol alters gene regulation such that it upregulates genes important for export of triacylglycerol in the hepatocyte by the hepatocyte, and it reduces the expression of genes involved in reesterification of fatty acids into triacylglycerol and genes involved in the pro-inflammatory response. Now, we decided to readdress this question with an experiment that we completed recently. So we assigned 110 cows to one of 5 treatments. We used the same hepatic lipidosis induction protocol. So we had cows that were fed at Lipton for a period of 5 days, and then all the cows underwent a period of feed restriction to supply only half of the calories that they need at that stage of gestation and according to their maintenance needs. Then we applied one of 5 treatments, and we measured lots of things, including intake, and we took blood samples and liver samples at strategic times. So liver was collected on day 6 and 13. I'll show you the data of day 13. And we took blood samples throughout the experiment. So the treatments were a negative control, no supplemental choline as rumen-protect choline, or 12.9 grams of choline ion as rumen-protect choline from one of 2 products. So a product with low choline chloride, and within this product, we doubled the dose of choline ion to 25.8 grams per day. And then we did the same, but with a product that had a high concentration of choline chloride. For the sake of time, I want to focus on the effect of choline, which are these 4 treatments against the control, and the effect of dose of choline, which is 12.9 to 25.8 grams being fed per day. So just to show you that we cause massive lipomobilization, this is the body weight change in 9-day period. Cows lost 2 kilograms per day. They lost body condition, and by losing tissue, the marker that we used was fatty acid concentration in blood, and you can see that there's massive lipomobilization and there's increase in ketogenesis. The important point here is it didn't matter what treatment they were assigned to. They all had marked increase in fatty acid and blood ketone. So what I'm going to show to you here is independent of the level of lipomobilization, which was very similar among all treatments. So here is the dry matter intake and net energy intake of the cows and the net energy balance during the feed restriction period. So these cows were deficient in 10 megacalories per day. Okay, so our treatment protocol induced negative energy balance as expected because we limit fed cows. And by doing that, we increase triacylglycerol in the hepatic tissue. So here's the concentration on day 13 during the period of feed restriction. And we can clearly see that we have high triacylglycerol concentration on the hepatic tissue on a wet basis, but the concentrations become smaller. They are reduced when cows were fed choline in the diet. And you can see that the reduction is greater when they are fed a larger dose from 12.9 to 25.8. Whether we look on hepatic triacylglycerol on as-is basis or on dry matter basis, we see exactly the same response. We see a decrease by feeding choline, and we see a greater decrease as we increase the dose of choline. When we look at glycogen, we see the opposite effect, which is a very positive effect. We see an increase in glycogen by feeding choline, and this increment is greater when we feed a larger dose of choline, such that if we look at the ratio of triacylglycerol to glycogen, which has been proposed as a marker of fatty liver, that ratio is much larger for cows in the control than in the choline supplemented group, and the ratio becomes further smaller when the cows receive a larger dose of choline. So this data corroborate with data from Reinaldo Cook, the Wisconsin experiment, as well as the data from Marcus Zenobi, the Florida experiment that I showed before. So I think there's very strong information that choline has lipotropic effects. It reduces the risk of fatty liver in cows when they undergo periods of negative nutrient balance. Now, in a subsequent experiment, we apply exactly the same treatment protocol. So we had feed restriction in those cows for a period of 9 days. And then on day 19 of the experiment, we dose cows with this non-ionic detergent to coat lipoprotein particles, such that we prevent lipoprotein lipase from hydrolyzing very low density triglycerides and very low density lipoprotein. So we look at the rate of accumulation of triacylglycerols. Okay. So this is what we did. We assigned cows to one of 3 treatments. So we had 33 cows. They were about 230 days of gestation. They were dry cows, and we assigned them to control, and then to one of 2 types or products containing choline ion or choline chloride, and the doses of choline ion that we fed were exactly 25.8 in both products. So for the sake of this conversation here today, I want to focus on the effect of supplementing choline and not necessarily on the effect of product. So the idea here is to prevent hydrolysis of triglycerides and VLDL particles in plasma or in serum, such that we can look at accumulation, and accumulation reflects the rate of hepatic secretion of triacylglycerol. And then at 6 hours after we dose this non-ionic detergent here, we collect lymph tissue from the superficial lymphatic vessels in the back of the udder. Okay, so this is what we found. This is the concentration of triacylglycerol in serum over time, over a period of 12 hours. And you can see that the accumulation is greater in the dashed lines than in the continuous line. So much so that we have a treatment by time interaction. The dashed lines represent the cows that received 25.8 grams of rumen-protected choline. So when we analyze the area under the curve, we can clearly see the cows supplemented with rumen-protected choline have a greater accumulation of triacylglycerol, meaning that they have a greater secretion of hepatic VLDL, which is the vehicle to export triacylglycerols from the liver. And we think this is part of the mechanism for reduction in fatty liver in cows, as it is the mechanism for reduction of fatty liver when choline is supplemented to monogastric animals. So again, cows seem to behave very similar to monogastric species, and choline clearly shows an increased ability to export triacylglycerol, which would reduce fatty liver in cows. Now, when we evaluate the lymph composition in those cows, so these are the cows in green that receive rumen-protected choline. The cows in red are those that were controlled. We have 30 cows that we sample lymph, that we were able to collect lymph fluid. as we go from blue to brown or red, the concentration of that analyte increase in the lymphatic fluid. And here are the analytes that were differentially concentrated in the hepatic, in the lymphatic fluid of cows. So you can see that they included diglycerides, they included triglycerides with different fatty acid composition. They include amino acids such as isoleucine, histidine, alanine, phenylalanine, but they also included choline. So we can see that the concentration of choline in lymphatic fluid increased as we go from right to left. And as I go from right to left, I go from the control cows that are clustered here to the cows supplemented with rumen protect choline. So we believe that by supplying choline in the diet as rumen protect choline, this choline is being absorbed and it's being incorporated into the cells, but also is being transferred either to the bloodstream by the portal system, but also being transferred to the lymphatic system, either through recirculation or by being taken up by the lacteals in the intestine. Now, I made the case that choline is lipotropic, so this should reflect into benefits to cows during lactation. So let me show you data from a meta-analysis in which we look at the effects of dose of choline that was supplemented in the diet of transition cows on subsequent postpartum lactation performance and health. So to be part of the meta-analysis, the experiment had to have a control treatment in which cows received 0 grams of choline ion, and they had to have a treatment that cows were fed rumen-protected choline starting prepartum. And here you can see the amounts of choline ion that were fed to cows starting prepartum. So all the way from 5.6 grams per day to as much as 25.6 grams per day of choline ion. You can see also that most experiments, most treatment means that we collect for the meta-analysis were in this range here of 9 to 13 grams because most experiments use 12.9 grams feeding rate for cows per day. Okay, so this is one of the responses that we evaluate is energy-corrected milk. And the way you read this figure, this is a forest plot. Each dot represents a comparison in the meta-analysis. The references of the experiments are listed here on the left. When the point estimate falls to the left of the central line, it tells you that the response was reduced by the intervention. And the comparison here is always rumen protect choline against the control, which is 0 grams of supplemental rumen protected choline. When the dot falls to the right, it means that the response was increased. The gray square represents the weight of that comparison in meta-analysis, which is listed here. And this is the 95% confidence limit, which is also listed here. And you can see that most point estimates are to the right of the central line. In fact, this is the overall response from the mixed model, and it shows an increment in energy-correct milk of 0.38 standard deviations, which represents 2.18 kilograms in this data set. So, but one important aspect of this is that there is a lot of external validity to this data because most points fall to the right of the line, meaning that in different experiments, different farms, different management, different diets, choline increased the response. In other words, energy-correct milk. But it also showed that there is heterogeneity in the level of response. Okay, so the effect of choline is dependent on other factors. Okay, and that's what we explored here. One of which is the dose of choline fed. So here are the controlled treatments. This is the response to level of feeding. This is the overall effect, which is the central line. And you can see as the dose increased from 0 to 25.6 grams per day, there was an increase in milk yield, and there was an increase in energy-correct milk yield, which is this linear effect of choline in the diet, of dose of choline. But there was also an interaction between dose of choline and concentration of metabolized methionine as percent of the MP of the diet postpartum, which was something that we explored. So let me show you what this interaction here looked like. So This is the metabolized methionine as percentage of the metabolized protein of the diet. So we had experiments. We put all this data into the National Research Council software of 2001. So the predictions are all based on the NRC 2001, which probably predicts a little less methionine supply than other models such as the CNCPS. And you can see if we look at 0 grams of choline ion compared with 12 grams of choline ion supplemented, we can see that in diets that are low in methionine, there is a substantial increase in milk yield. And in diets that are high in methionine, there is also an increase in milk yield. So in both cases, we see an increase in performance, but the increment becomes smaller as we increase the supply of metabolized methionine. If we look at energy-correct milk, it's the same type of response. We see here a 2.4 kilogram increase with 12 grams. If we go to the maximal level, we see a 4 kilogram increase in milk. If we get to diets that are very high in methionine, 2.4, 2.5, the increment smaller but still present, okay, 1 to 2 kilograms more. So the point I'm trying to make here is that although biochemistry has an interplay, okay, obviously this is showing that both the response of choline is dependent on methionine, but there seems to be an additive effect that even in the presence of high methionine, we still capture a response in milk yield and fat-correct milk yield. Now let me show you the effects on health of cows. An important aspect is not all we had 21 experiments reported for the meta-analysis, but only 11 or 12 of them report disease incidence in cows. Okay, so the data set is not as robust for disease as it is for milk yield. And this is probably why for many of the disease we were unable to detect any impact of choline. But for the risk of retained placenta and the risk of mastitis, we saw a reduction with supplementing choline compared to control. And the reason why I'm showing you here choline as a category is because almost every experiment that reported disease fed 12.9 grams of choline ion as rumen-protected choline. So here I make the case that there is a lot of external validity for the production response when cows receive choline. They receive by increasing milk yield and energy-correct milk, okay? But they also respond by reducing the risk of some disease postpartum. Now, in 2 experiments that we conducted here at the University of Florida, cows were fed rumen-protected choline starting at 255 days of gestation and stopping at exactly 21 days postpartum. So they were fed during the transition period. And you can clearly see that supplementing choline, which is the close symbols here, increase milk production. But more importantly, the increment was extended well above the period of supplementation, up to 40 weeks in the experiment of Marcus Zanobi and up to 25 weeks in the experiment by Juan Bolatti. So in both cases, we saw a 2 kilogram increase in milk yield for a period of 25 to 40 weeks, although cows were supplemented for only 3 weeks only 6 weeks during the transition period. And I think that probably part of this extended benefit comes back to the fact that during the transition period, we have the smallest concentrations of choline or phosphatidylcholine in plasma of cows, and probably by supplying choline, we increase the supply of substrate for phospholipid synthesis. And this is right at the time in which proliferation of mammary cells are upregulated. So this is the transition period here, and this is activity of choline kinase, that enzyme that phosphorylates choline into phosphocholine. And this enzyme is highly active in the mammary epithelium, such that when there is proliferation of tissue, the enzyme is upregulated. So not only the gene is upregulated, the protein is, and the activity of that protein is upregulated. And this enzyme here is a marker for cell proliferation, in particular during breast cancer in humans. So it is possible that by supplying more choline, we increase the abundance of phosphatidylcholine that supplies the mammary gland with substrate for synthesis of phospholipids, and this upregulate the activity of choline kinase because we provide more choline, and this may stimulate mammary cell proliferation that results in this extended benefit for lactation performance. Lastly, I want to touch on this concept that probably every cow benefits from choline, and the example that I'm going to use is body condition score. So we went back to our data in 2 experiments that has about 230 cows, and we asked the question, does body condition score of cows when they enter the close-up period or the close-up pen affect how they respond to choline? So this is the distribution of body condition score of cows in the control treatment in Marcus Zenobi's experiment and in the choline treatment, and the same for Juan Bolati's experiment. So you can see that we have cows that have low body condition or moderate body condition, and we have fat cows that are over-conditioned. Okay? So the question we ask, do these cows that are low body condition respond as well as these cows that have high body condition to supplemental choline during the transition period? And I'll show you the distribution of the body condition score. You can see that we have a wide spectrum of body condition all the way from less than 3 to more than 4 in both treatments. And there's about 115 cows in each group here. And this is the milk yield and the energy correct milk yield for those cows. And the way you look at this figure is that you can clearly see that as I move from the blue line to the orange line, there is an increase in milk and there is an increase in energy correct milk of about 2 kilograms per day. And this increase is independent on body condition score. So let me remove the dots which represents every cow. So you can see here no interaction between supplementing rumen protect choline and the body condition score of the cow when she entered the close-up pen. So skinny cows respond to choline with more milk and energy correct milk. Fat cows respond to choline with more milk and more fat correct milk. So just to finalize, I want to present some potential mechanisms for improved production and health when cows receive choline. Obviously, the first one is this classic mechanism that choline provides substrate for synthesis of phosphatidylcholine, which is important to synthesize very low density lipoprotein particles that export triacylglycerol from the liver, as it's been shown in Usman Arshad's recent work. And this probably explains the reduction in fatty liver in cows that I showed you very consistently from 3 experiments. By supplying choline to choline-deficient diets, we enhance the synthesis of chylomicrons, which is illustrated here in laboratory animals based on the concentration of polypoprotein B48, which is a marker for chylomicrons. And chylomicrons are lipoproteins that facilitate the transport of absorbed lipids in the gastrointestinal tract to be delivered to other tissues. So this would make the case that by supplying choline to animals that are deficient, enhances the ability to transport absorbed nutrients from the gastrointestinal tract to be delivered to other tissues. And in fact, when Marcus Zenobi supplemented choline to cows and dosed them with fat, he showed that choline supplemented cows had increased concentrations of triacylglycerol in plasma compared to control, suggesting that probably absorption and transport of lipids is enhanced in the gastrointestinal tract of those cows. We are very aware of the fact that cows are predisposed to this disruption in gut structure that results in leaky gut, and deficiency of phosphatidylcholine is a risk factor for leaky gut. Phosphatidylcholine is important for epithelial cell integrity because it's part of synthesis of cell membrane, and the structure of these gap junctions is enhanced when we have intact enterocytes. So when animals are deficient in phospholipids, it's possible that leaky gut that might be happening during transition period may be exacerbated. Also, phosphatidylcholine has anti-inflammatory effects. It attenuates the pro-inflammatory response, which probably enhances the integrity of the gastrointestinal tract and improves liver health by attenuating a pro-inflammatory response that might be induced by accumulation of lipids. But phosphatidylcholine has or choline in general has some other very cool effects. And one of them is this experiment here in which they bred mice to have microbes in the gastrointestinal tract that are unable to destroy dietary choline into these 3-methylamine oxides. So they had the wild animals that had microbes that destroy choline. Therefore, the supply of choline to the offsprings, to the offspring of those dams was limited. Whereas in mice that they bred to have this transgenic microflora that doesn't express the enzymes that degrade choline in trimethylamine, they would have increased supply of choline to the dams as well as to the offspring. And what they observed was that by supplying more of these methyl donors, there was increased DNA methylation. So methylation of histones in the DNA were enhanced. And by methylating, you suppress opening of chromatin. So there is less gene expression. And some of the genes that were suppressed, they were involved with reduction in adiposity, reduction in metabolic disease, and changes in behavior of the animals, so less anxiety. So by providing more choline through the less action of these degrading enzymes in the gastrointestinal tract, they change the phenotype of the offspring of those animals, which is a very cool response. And lastly, I want to leave you with this concept that there is this enzyme, choline kinase, that phosphorylates choline into phosphocholine. And this provides substrate for the novo synthesis of phosphatidylcholine in all cells. But this is probably very important to mammary epithelial cells that are replicating during the onset of lactation. And perhaps by supplying choline, we influence the activity of this enzyme in addition to providing more substrate. And this enhances cell proliferation, maintains cell membrane integrity. And this may explain the fact that cows keep responding with additional milk yield and energy correct milk yield well beyond the period of choline supplementation. So I just want to conclude that I think it's unquestionable that choline fits the criteria of required nutrient in cattle, as it does for other species. It reduces triacylglycerol infiltration in the liver by enhancing export of triacylglycerol as lipoprotein. It benefits lactation and reduces the risk of disease, all important criteria that makes a nutrient required. The response is influenced by the supply of metabolized methionine and by the dose of choline fed, but is still present even in high methionine diets. The benefits are observed regardless of body condition score, and I think we need to be sensitive to this. It's not just the fat cow that responds. The skinny cow also responds to that. Okay? So it should not be targeted only to overconditioned cows. It should be targeted at this point to the entire population. And the mechanisms that justify improved productive performance, it may be related to increased nutrient intake. I didn't show you this data, but there's some data to substantiate that. Perhaps improved nutrient absorption and integrity of the gastrointestinal tract. Obviously, there is less fatty liver. There is reduction in clinical disease. And perhaps we have this other non-classical mechanisms such as reduced inflammatory response and improved memory cell proliferation.
Well, Clay, that's going to be last call. Dr. Santos covered a lot of ground and many research studies. In your mind, what is the biggest takeaways for nutritionists and dairy producers from his talk?
Our last call question is sponsored by AminoSure XM Precision Release Methionine, the next generation in amino acid balancing. With AminoSure XM, you can save up to 5 cents per cow per day on your methionine investment. Try it today and receive an additional 2.5 cents per cow per day savings with Balchem's limited time rebate offer. Contact your Balchem representative to learn more.
Probably the biggest thing was, he spent quite a bit of time, discussing the most recent meta-analysis that was done out of his lab, published back in 2020. Showing the consistency of response to rumen-protected choline across all these transition cow studies, and also the fact that you get a response to rumen-protected choline no matter what the production level of the herd. It's a very consistent response. That really ranged from about 45 pounds of milk in the control cows at the low end, and up to 100 pounds per cow in these control treatments. So nice responses across a very wide range of production levels. He discussed some potential mechanisms of how choline may work in the transition cow. Some of these we know, some are still theories at this point. We know, we clearly know that it has a positive effect on liver health, and that really has lots of positive effects for this fresh cow. But he discussed some other potential modes of action that quite frankly we're studying, trying to learn more about. We as we're running, continue to run research studies with Reassure, we learn something new every time. So he was alluding to some different potential modes of action that are there with choline in the transition cow. And really, one of the last things he talked about was based off of actually another small meta-analysis that they did, looking at whether or not the response to rumen-protected choline is related to body condition score of these dry cows, because there certainly has been a thought in the industry that maybe rumen-protected choline is only for fat cows, and that's not true. There is they looked at data from different studies. It was over 300 cows total, and there was absolutely no correlation between prepartum body condition score and the production response to rumen-protected choline.
Yeah.
So really shooting down that dogma that's been out there in some circles.
Yeah. So Clay, one of the things I found interesting in the talk was the discussion around body condition score and the feeding of rumen-protected choline. Could you kind of touch on that for a moment?
Yeah. So they Jose published a paper, I think it was back in 2021 now, at looking at is there a relationship between prepartum body condition score and the response to rumen-protected choline? Because there's been a dogma out there that only fat cows respond to rumen-protected choline supplementation, and that was absolutely not true. There was no correlation between prepartum body condition score and the response to rumen-protected choline. So whether they were thinner cows or heavier cows, they all responded equally with increased milk and energy-corrected milk yield.
Great summary, Clay. Remember, this is just one in a series of 4 podcasts around the new revelations in transition cow nutrition. Be sure to tune in for the rest of the series. And if you haven't listened to the first one, go back and listen to the first one by Dr. Mike Van Amberg. The next podcast will take us into a deeper dive into some of the newest transition cow nutrition research coming out of Michigan State University with Dr. Barry Bradford. So we'll look forward to seeing you there. To all of our loyal listeners, thank you again for coming along for more than 60 episodes and sticking with us as we explore more topics. We hope to see you next time here at the Real Science Exchange, where it's always happy hour and you're always among friends.
We'd love to hear your comments or ideas for topics and guests. So please reach out via email to anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars continues with ruminant-focused topics on the first Tuesday of every month, monogastric-focused topics on the second Tuesday of each month, and quarterly topics for the companion animal segment. Visit balchem.com/realscience to see the latest schedule and to register for upcoming webinars.